Namaste students. Today we are starting with our chapter 6 in class 12th. The chapter is about the molecular basis of inheritance, and today we are covering the topic that is the structure of a polynucleotide chain. This is the topic before DNA. We are taking this a polynucleotide chain. Now going to the history of the discovery of the nucleic acid. The name comes the Friedrich Mischer. He was the scientist while working with the pus cells. He found some acidic substance in the nucleus and he named it as a nuclein. Thus, nucleic acids were discovered. Nucleic acids has an acidic behavior because of the presence of a phosphate group. It is an important point, that is the acidic behavior due to phosphate group. Now, coming to the types of the nucleic acids, we have two types of nucleic acids, that is DNA and RNA. DNA is deoxyribonucleic acid and RNA it is the ribonucleic acids. Now, these two molecules are actually the polymers. Now, we can come across the polymers, that means it is made up of a number of monomer units. The structure, it is a polynucleotide chain. We call it as a polynucleotide chain. When we say a polynucleotide chain, that is poly means many nucleotides. So, the monomer unit is the nucleotide. Here the nucleotide is the monomer unit. And the nucleotide, now we see the components of the nucleotide. A nucleotide has three components, that is a sugar, a nitrogenous base and a phosphate. These are the three components of a nucleotide. Now we see one by one all the three components. The first one is a sugar. Now the sugar we come, here it is a pentose sugar. Pentose, as the name indicates, it is a 5 carbon sugar. Now we draw ring structure of this sugar. The 5 carbons are present in a manner, that is 1, 2, 3 and 4. You must remember the position of these carbons, the numbering of these carbons, that is it is important in the bonding. So, sugar is of two types, deoxyribose and ribose. Ribose sugar, as the name indicates, is present in RNA. It is a component of RNA, while deoxyribose is a component of DNA. Now, because of this sugar only, the naming is so. See, ribonucleic acid because of the presence of ribose sugar, deoxyribonucleic acid because of the presence of deoxyribose sugar. Now, there is a slight difference between the two sugars, that is, you can see at carbon number 2. Carbon number 2 has a functional group OH in case of ribose, where it is H in case of DNA or deoxyribose. So, there is a slight difference. Here two functional groups are present, while here only one functional group is present. This is deoxy. You can resemble in the name, D is the deficiency, oxygen, oxy for oxygen. So, there is a deficiency of a single oxygen in deoxyribose sugar. So, this is all about the sugar. Now, we come to the nitrogenous bases. Now, nitrogenous bases are the ringed compounds. Ringed structures, these are of two types, purines and pyrimidines. Now, come to the purines. Purines are of two types, adenine and guanine. In the further studies, we will take it as A and G. And pyrimidines are of three types, that is cytosine C, thymine T and uracil U. So, there are three types of pyrimidines and two types of purines. Now, here is again comes a difference, is the uracil. Uracil is present in RNA. Uracil is present in RNA in place of thymine. So, both RNA and DNA has only 4 nitrogenous bases. In DNA it is A, G, C and T, while in RNA it is A, G, C and U. So, these are the 4 bases. Next comes a phosphate group. A phosphate group is also present in the nucleotide. So, these are the 3 components of a nucleotide. Now, we see how these components are bonded together. See, we say a nucleotide, a monomer unit is a nucleotide. A nucleotide is formed of a nucleoside plus a phosphate group. See, a phosphate and a nucleoside. Then what is a nucleoside? Nucleoside is a sugar and base. See, when sugar and base combines, it forms a nucleoside. And when this nucleoside combines with a phosphate, it forms a nucleotide. So, this is a simple bonding. Now, we see the names of the bond. See, when the sugar is bonded with the base, the name of the bond is the N-glycosidic linkage. Here the name is of importance, the bond name. It is N-glycosidic linkage between the sugar and the base, whereas when this nucleoside is bonded with phosphate, the bond is a phosphoester bond. Remember it is a phosphoester bond, and ester linkage is present between a nucleoside and a phosphate group. Now, just we come across this sugar, the position where these are bonded. From the carbon number 1, a base is bonded. Base is linked always to the first carbon of this sugar, the carbon number 1 of the sugar. Then the phosphate group is always bonded with the 5 number carbon of the sugar. This numbering is important. Base is always bonded to the carbon number 1, whereas phosphate is always bonded to the carbon number 5 of the sugar. So, this forms a nucleotide having a phosphate group, a sugar and a base. Now, this nucleotide is again bonded with the next nucleotide. So, the two nucleotides are bonded together. Two nucleotides are bonded together with a bond known as the phosphodiester linkage. A question often comes, then how the two nucleotides are bonded together? It is bonded together with a bond that is known as the phosphodiester linkage. So, here this phosphate is bonded to the third carbon of the next sugar, and this is how a complete sequence of monomers forms, and it is known as the polynucleotide chain. Now, remember it, when we give the structure of a polynucleotide chain, it is common for DNA and RNA both. So, a polynucleotide chain is common for DNA and RNA. If the question comes for RNA, you can mention the same thing. Just remember the base and the sugar, the difference in the sugar and the base. So, in the further section, we will study about the DNA, the double helical model of DNA. Here it is the polynucleotide chain. This knowledge is coming in the next session also. Thank you. So, this was the structure of DNA. Thank you.